welcome back to Science of Medicine. Today I would like to, in this video, talk about the endocrine pancreas functions, principally that of the beta cells producing insulin. You may remember when we talked about the gastrointestinal tract that we discussed in detail about the exocrine pancreas, that in addition to secreting bicarbonate, also secreted a number of enzymes that were necessary for continuing the digestion of things that pass through the duodenum. The endocrine pancreas is a much smaller amount of tissue compared to the exocrine pancreas, and it has a different set of functions. So let's talk about those. So if I remind myself of the pancreas, remember that the head of the pancreas is nestled within the duodenum, and it has these exocrine pancreatic ductal work that goes through the sphincter of Oddi into the duodenum, and that's where I dump my digestive enzymes as well as my bicarbonate to neutralize my hydrochloric acid that's coming out of the pylorus. But I also have these sets of tissues that are highly vascular. And these are the endocrine portions. They're called islets of Langerhans. And they have different cell types. Beta cells, which I said before, will synthesize actually proinsulin, which will then be cleaved into insulin. Alpha cells, which synthesize and release glucagon. D and F cells that then secrete somatostatin and pancreatic polypeptide, which I'll very briefly touch on. Now, the most talked about portion of the endocrine pancreas is that associated with the beta cells, because the diseases that we have associated with that is diabetes mellitus, is a very common disease for humans. And so functioning within the beta cells and insulin becomes critical to this. It's also the largest mass of the islets of Langerhans, about 60% of the cells. Now, they secrete proinsulin, and proinsulin, which I'll talk about in a second, will be cleaved to produce insulin and C-peptide, and then also amylin. Now, amylin is being worked on in terms of what all of its functions are. It does things related to gastric emptying, with respect to you feeling satiated after you've eaten a meal. But one of the things that I wanted to point out to you about insulin is that because the beta cells secrete proinsulin, and insulin then is cleaved to form this C-peptide in insulin, the insulins that we actually give for people that have diabetes mellitus does not have the C-peptide, it just has insulin. And we'll talk about the reason for that in a few minutes. Now, the alpha cells, which are about 25% of the cells, secrete glucagon. And glucagon kind of does the opposite of what insulin does. It'll increase synthesis and release of glucose in the liver. And in fact, for people that have very severe hypoglycemic episodes that can't take anything orally, we use glucagon as a treatment for that severe hypoglycemia. D cells, which are the next most abundant cells, secrete somatostatin, and I'll touch upon that later. And then F cells secrete pancreatic polypeptide, the function of which we're not exactly clear about, so I won't mention beyond this. Now, as I've mentioned, the beta cells synthesize proinsulin, and proinsulin is cleaved into insulin plus C-peptide. And as I mentioned before, the insulin that is injected by people with diabetes mellitus does not have C-peptide, and that'll be important because it's one of the ways that we can determine whether a person's hypoglycemia is due to secreted insulin, which is coming out with C-peptide, or from iatrogenic, or that person giving themselves insulin, which does not have C-peptide. Now, what does insulin do? It does a lot. It has many cellular effects. In muscle and fat cells, it will be associated with facilitation of uptake of glucose into the cells. Now, in neurons, glucose is freely permeable, so you do not need any insulin binding to get glucose to be taken up. But in muscle cells and in fat cells, you do need insulin to facilitate this. It also allows for increased permeability of various fat and muscle cells to amino acids, to various ions, and ups enzymatic activity. So insulin is necessary when you're really trying to use these muscles to facilitate their use. In the liver, it inhibits an enzyme that breaks down glycogen, because if you have lots and lots of glucose, you don't need to be breaking down glycogen into glucose. It does increase the amount of hepatocyte uptake of glucose that then can be synthesized into glycogen or converted into fatty acids and glycerol. On the protein and fat side, it inhibits the catabolism of proteins, which can go through gluconeogenesis and promotes protein synthesis. In fat cells, it inhibits lipase inside the cells and promotes that conversion of glucose that you have lying around or excess into fat. Now, if you look at insulin secretion by these beta cells and you track that with glucose in the blood, they track perfectly. This graph actually shows you a temporal level of blood glucose levels in red and insulin levels in blue. And you can see that as the glucose level goes up, the insulin level matches that. And this is designed to keep your blood glucose into a very tight range. Under normal conditions, when you are fasting, your blood glucose is kept between 70 and 100 milligrams per deciliter. When you eat, assuming you do not have diabetes mellitus, your glucose could rise really as much, but not more than 140 milligrams per deciliter, and many times it's down closer towards that normal level. And that's because of this secretion of insulin, which will track the increase in blood glucose and bring it down. And you can see for this person, for each meal, as their glucose rose, so did their insulin levels. But I sometimes have trouble. And there are two sort of generic things, as we talked about before, for any hormonal disorder where you have too much or too little. Too much insulin can be from one of two causes. Very, very rarely you will have a tumor called an insulinoma, which secretes insulin, not linked at all to what your blood glucose is. This is exceptionally rare. More commonly, sometimes people will inject themselves with insulin. Now remember, this insulin that is injected does not contain C-peptide, and it's a way that we can actually determine or distinguish between people that are injecting themselves with insulin and those that might have an insulinoma. But these are rare diseases, although injecting yourself is not as rare as an insulinoma. The most common diseases related to insulin problems are deficiencies. And the deficiency of insulin is called diabetes mellitus. And there are many types. I will break in and discuss two types, which are sort of extremes. Type 1 diabetes mellitus is where the person lacks the ability to secrete insulin. And this was a fatal disease up until two medical students actually doing research in 1921 actually isolated and identified insulin as this necessary component and then gave it to a patient the next year, and then that basically allowed that person to live. Up until that time, it was fatal uniformly to have type 1 diabetes mellitus, where the beta cells were basically attacked and destroyed in the body. It is still true that if you have type 1 diabetes mellitus, you must give that person insulin. This can be either from autoimmune destruction of the beta cells, or it can also be in people that have harmed or have a pancreas that has been damaged. Alcohol abuse will actually do this. You will have to give that person insulin because they're unable to secrete it. The more common disease, more prevalent, is type 2 diabetes mellitus. And this is where the person does secrete insulin, but it is insufficient to keep up with the needs of the body. And we normally frame this in terms of it being a resistance to the effects of insulin. And this requires more insulin or something to deal with the blood glucose levels, or to try to decrease them, or to decrease the resistance to the effects of insulin. And there are other types of diabetes mellitus, but these are the main ones. Now, as I mentioned before, the alpha cells will make glucagon, and this is a polypeptide. It's actually synthesizes proglucagon, cleaved to give that glucagon. You also get these molecules which are called incretins. GLP-1 and GLP-2 are examples of incretins. And in fact, there are medications that are used for type 2 diabetics that act on these GLP-1. Now, the function of glucagon is to basically raise the blood glucose. So it's going to increase the rate of glycogen breakdown to glucose and increase gluconeogenesis. It is also going to activate lipase and in cells and inhibit the storage of fats and triglycerides. And its effector is blood glucose, just like insulin, but now it's going in the opposite direction. When blood glucose levels drop, it will stimulate the production of more glucose. It will mobilize glucose. Also, exercise and stress will cause this to actually be secreted too. As I mentioned before, in a patient who would have very, very low blood glucose levels, which will be fatal if they're too low, if that person cannot take anything orally, we have a formulation that we can give glucagon as an injectable, and this will help raise their blood glucose levels. The last thing that I'll talk about is somatostatin. And so somatostatin suppresses insulin and glucagon secretion. It's kind of a regulatory. It will decrease the motility in the stomach and the duodenum and the gallbladder, and decrease absorption in the GI tract. So I hope that this has given you an overview of the endocrine portion of the pancreas, and I will, as always, come back and talk with you a little bit more about the endocrine system. We'll talk about reproduction next. I hope that this has been helpful. You're also welcome to subscribe or leave comments. Otherwise, I'll see you soon in Science and Medicine.